So, hi all, and good afternoon, and welcome to CIDAI Masterclass that today is about finding a drug for your target. It is a problem. It is still a problem. Do you know that molecular modeling can play a key role in drug discovery and personalized medicine? I don't know about it, but let's discover a little bit more about it, and let's answer this question with Victor Guallar. Victor Guallar will be the speaker, today's speaker, and he will provide us good insights about this issue, about this molecular modeling. So, welcome everybody again. I am Ana Escoda, the Technological Transfer Manager at Barcelona Supercomputing Center, and I will introduce you the speaker, and in some way I will conduct the session. But first of all, let me provide you some insights about CIDAI. I know that most of you are aware about CIDAI activities and CIDAI objective, but let me remind you that CIDAI is a Catalan platform, and CIDAI is playing a key role. So CIDAI is a key element for the Catalonia artificial intelligence strategy. The CIDAI mission is to demonstrate the advantage that to speed up the adoption of innovative data exploitation and artificial intelligence technologies for both for science and for industrial and small and medium companies side. So which role Barcelona Supercomputing Center is playing in this CIDAI platform? So Barcelona Supercomputing Center is a core partner on CIDAI. We are enrolled in this platform since the beginning, 3 years, I guess that is 3 years ago. And you know that Barcelona Supercomputing Center is a research center. We are focused on some research lines from computer science research areas to life science research areas. And we are working on several tasks in order to increase the adoption of these technologies that are the ones who are developing, the ones that are a result of our research center. And our role in CIDAI is to provide you the project outputs based on artificial intelligence, and together, meaning together, research and industrial side, find a way in which you can adapt this in order to work for a better performing of let's say economic and industrial sector. So this is the objective of this masterclass. Today we are going to talk about Victor Guallar research lines. Victor Guallar's research lines are focused on theoretical model and biochemical and biophysical processes at molecular, atomic and electronic level. But before introduce he as a speaker, I want to remind you that if you are working on a small and medium company and you have ideas to develop some proof of concept based on artificial intelligence, or you, based on the insights that Victor Guallar are going to provide you, have the idea of can use or can adapt some of these project results into your, let's say, daily life in your company, please contact CIDAI via CIDAI platform, and we will provide you some information about how to proceed in order to join in some proof of concept or something like this. So today, as I said you at the beginning, the speaker will be Victor Guallar. Victor Guallar is an ICREA professor at BSc, and Victor is also a CSO at Nostrum Biodiscovery. Biodiscovery is the first spin-off at BSc, and is a company mainly focused on using these molecular modeling tools for helping pharma sector and others, and industrial sector, pharma sector on drug discovery, and industrial sector to develop some new moleculars, etc., that can provide a good performance at industrial level. So Victor Guayar is leading the Electronic and Atomic Protein Modeling Research Group at Barcelona Supercomputing Center. And in this master class, he will outline the major trends in this theoretical modeling and biochemical and biophysical processes at molecular, atomic, and electronic level. So Victor is going to explain the emerging trends on it, the key players on it, and the current and the future challenges in these areas. So Victor, the floor is yours, and in order to start the session, I want to ask you, so it's not necessary that you answer it right now, but finding a drug for your target isn't a problem anymore. So Victor, let's explain us a little bit about this. Thank you.
Thank you, Anna, and thank you, the organizers, also for this invitation. Can you hear me well, Anna? Just first, let's ask that.
We can hear you, Victor.
Okay, excellent. So let me see if I can start sharing. If you pass me the presentation, I will be sharing. So two of the main ingredients that have made this, we can even call revolution or breakthrough advance, are high performance computing and artificial intelligence, and along with some other ingredients, but certainly these two are permitting us to really say today that finding a drug, once you have a target, let me rephrase this question. If you have a well-defined target, finding a drug is now, in most of the cases, will be fast and very accurate. Let me frame the problem a little bit more. So you see, especially this talk will not go too much into detail, because I understand that the audience is diverse and that they probably don't have too much experience in drug discovery or biotechnology in general. But let me frame the problem. So we have a huge industry, which is the pharmacological industry, trying to find better cures, trying to find medicines that will fix some problems. And finding a drug that for a given disease is a very lengthy and expensive process. It's a process that takes, on the average, between 10 to 15 years. And it has a lot of different phases, like we see in here. One of them, the initial one, is the target validation. So we need to know who we are fighting against. So if you have headache, you can try to block some neurons that will give you this pain, sensation of pain, and therefore we went after trying to block in some neuron receptors, so the neurons don't transmit the signal. Or if you have cancer, you can try to accentuate some receptors that will induce the cell death for those particular cancer cells, etc. So once we have the target validation is here I put dotted lines because it can be super lengthy process, could take 10 years, 20 years, or not so much, but it's quite undetermined and it varies a lot. Once you have the target, then you go into the early phases, which is basically all the preclinical phases that are trying to find one molecule that will make the work, that will touch the given target, targets typically being proteins, will touch this protein and will block it or will accentuate it, depending on the mechanism that we are working. And that traditionally takes between from 2 to 4 years. I mean, here again, This is a very general number. There are exceptions, people who did it in much faster time, other people who probably took 5 years, but you find basically a molecule that needs to be then optimized a little bit for turning that molecule into a real drug, for example, no secondary effects, good administration properties, etc. And then after you have all that, within 3 to 5 years, now you go into clinical phases. All right. So that's altogether involves a lot of money and a lot of time, and it's very likely. So if you can make that much faster, if you can really make the finding the molecule in the preclinical is faster and much better, then you will have actually much more successes. Because one thing that I have to tell you is that even after all this time, very, very few molecules make it into the pharmacy. All right, it's to the drugstore. It's just 90% of the chances you will fail, and all this money that you have spent will be gone for nothing. Maybe you will get some knowledge, but it will not be transmitted into a given compound. So two things that have really enhanced that the screening, that finding molecules. One is supercomputing, and supercomputing meaning that we can do a lot of computation, big computations, but also that we can do medium computations, or not so big, but that might take some time, significantly faster. And here, of course, we are talking a lot about cloud computing. Pretty much every pharmaceutical company uses every day Amazon Cloud or Microsoft Azure or any other cloud provider. And why is that? Well, because they are very reliable, 24/7. They work all the time. They are scalable. So if now you need, if for a couple of days you don't need it, you don't have to be using it. Instead of that, if you have your own computers that you buy and you don't use it, this is money that you don't really you put to use, some of your expensive resources. So I guess it's on demand, it's scalable, you can use more or less, it is secure, and that's as you can imagine is extremely important. Developing a drug is hundreds to thousands of millions of euros. If that drug gets the market, that can be hundreds of millions of revenue as well for a company. And of course, all the information has to be very secure. And big cloud providers, Amazon, Microsoft, can provide the security. At least they can provide the security that they will pay you if they have some breach. So they could pay you the insurance for that. And also, it's personalized. We can tailor, even if those machines are going to be in the other side of the world, they can really tailor your space with your preference. That will be working with your preferred software with your technicians. It can be tailored for your needs. So that's quite nice. The other thing that has introduced the other component in the revolution I'm trying to show you here is AI, artificial intelligence. I'm going to be using artificial intelligence and machine learning indistinguishable. I know I shouldn't do that, but I guess we are corrupted a lot, and sometimes I will be referring to AI, when most of the time I'm referring to really machine learning. So finding algorithms that can really do search, optimize search much faster. Although AI certainly is coming now more into the game, even at our level of research. So I don't have to convince anyone that's a revolution. I mean, you're here because you are certainly interested in it, and probably everybody here has been playing with ChatGPT in the last months. I'm certainly blown away within the limit, within with criticism, and I'm going to be quite critical today with AI at some point. So you will see that. But I would say that 4 years ago, none of my research projects had AI in it. 3 years ago, probably none of them. 3 years ago we started, maybe 4 years ago, and now absolutely every project, 90% of the projects have some AI component, or we are thinking to introduce one very soon. So it has changed our life. And here I'm adding, you will see here in the right, but it still has not meet the expectations in drug discovery. And that's a reality, what I'm going to be showing to you today. It still has not been able to perform, and you will see that's a big component of the revolution, but it's alone, it's not enough yet. That's what I'm going to be going through. So here I just summarize a little bit of the early phase drug discovery process of target identification, screening. So once you have your target, finding a molecule that can interact with your target, one or a diverse set of molecules, the more the better. Then you have to optimize this set of molecules to come out with 1 to 2 to 3 candidates that maybe you might move into clinical stages. First some animal models, and then if that works well, different phases of clinical stages. So what I'm going to be showing, I'm going to be trying to interrogate these different aspects, and I'm going to be trying to tell you what we have been doing up to now and what is expected to be doing. And mostly we'll be focusing on the screening, but I will say something about the other aspects as well. So target identification, as I told you, it's a very complex aspect. It might take 20 years to really identify a target. It's very complex. So you need to identify first phenotypes, you know, why this, what is causing this disease, you know, understand the molecular mechanism of that disease, and understand why, when you will be touching some molecule, some protein, you will be curing that. So that has to be a very clear picture, that by touching these, you're curing that, but you are not making worse in some other sense, no, that's not toxic, etc. And there's a lot of ingredients that play in this field. So proteomics, phenotypic data, clinical data, you know, there's a lot of variables in order to decide what will be our target. And of course, as you can imagine, when we have all these variables and we want to make some sense of them, AI is a great tool. And we are expecting that the language models will be a big asset in here as well, in order to not only analyze some data, but to have this dialogue with scientists that will help the scientists to interpret data and to provide answers that, you know, maybe it will still need some refinement from the scientists, but in this dialogue and the potential of processing all that data, I mean, certainly it's going to be, I expect it to be a little revolution in the next 2 to 3 years in this particular aspect of drug design. And because what I'm going to try to convince you today, because I'm going to try to convince you that once you have the target, it is a done deal problem. Now the main aspect of drug design of the entire pharmacology will turn into target identification. So I think it will be the essential, the key aspect from now on. So once you have identified this target, and in here I expect AI to be a major player already is a major player, but it will be probably the player in the next years. We have other artificial intelligence techniques that have allow us to move from the knowledge of that target that typically is at the sequence level. So we have a protein, we can sequence this protein, and then we have which amino acids compose this protein. So another machine learning tool has been able to transform that sequence into a structure. That's the one that probably all of you have here, which is AlphaFold. Others as well, so it's not the only deep learning, but it was the first one. And that was done by DeepMind, participation of Google. And we use it in the lab on the hundreds or thousands of times per week now. It's a major player of, and you will see that in applications I'm going to be portraying. You might say, and people are saying, it's probably the biggest biotechnological AI advance in the history of AI, and that, you know, we believe that these people will get the Nobel Prize soon, and probably that's why the code is free to use and it's available. So having said that, we just published around 3 or 4 months ago work with RA Group in Boston, where we were benchmarking, and we are not the only ones. Other people have done that. So in the same issue, actually, there is another paper from Schrödinger doing basically the same things and coming out with same arguments, conclusions, basically. So these structures provided from AI, provided from AlphaFold, are not ready yet to do virtual screening to interrogate using them directly for drug candidates. I'll show you what the virtual screening process is, but if you use the structures as they are, you get really poor enrichment factors. Enrichment factor meaning that on the top molecules that you select for after you screen, there are real true molecules that are inhibitors of this protein. So that doesn't happen. And here you see some image already, one of the first images between a real close look at an active site of a protein. So the protein will be everything that's in pink and green. Here the backbone of the protein and some side chains, some amino acids of the protein, and how they interact with one of these molecules. And because the prediction of AlphaFold does not take into account the ligand, what it tends to do is to close a little bit this active site. You can see in gray is the crystal, and in pink and in green are the predictions. The predictions try to close the empty pocket, and that closeness of the empty pocket does not allow you to do a good job in virtual screening. So you need to do some molecular modeling of those structures, which is fine because you can do it. It's not a problem. But for people who are not proficient in this, I mean, they should be advised not to use the structures directly as they come out of AlphaFold predictor. So let's go into virtual screening, which will be the main topic today. So once you have that target, here is that target is this blue thing, this cloud. It's not real that we should be looking at more at this atomic level. But once we have that target, the targets, these are proteins. Proteins make a function in our body, and these functions can be, from a metabolomic to transmitting signalings to everything. And typically these functions happen because there is a substrate that binds in the active site. So for example, here, this half star symbolizes the active site, and there's a substrate that does bind here, and then it activates the mechanism. Or for example, there is another protein that interacts with this protein, etc., etc. So the traditional way of pharmacology is to find molecules, drugs, that will bind into this active site and will block it from its action. So it will be very different from the real substrate. So it will not activate the enzyme for reaction. It will not allow it to do its function. And by doing that, we will blocking that, and then we will be because our target identification has told us very clearly that by blocking these, we could cure this disease. So that would be great for us. So now the main problem is to find this needle in haze and find this molecule that will bind in here, which is a very, very, very complex problem because there's a lot of factors in it. All right, there's a lot of interactions. It's a quite complex problem, and that's why it takes 3 to 4 years, 2 to 4 years, again, generally speaking, to find a very, very good molecule that will be doing this work and will be capable of being transformed into a drug to be sold in the drugstore. How we done this from now? What's a traditional way of doing this? So the traditional way of doing this is using these mathematical functions, which we call four fields, which are very simple functions. You can see here this quadratic term. So these are rather simple functions. And these functions describe the energy, or describe the energy landscape, or a view of the molecules, like this ball and a stick. So molecules, atoms will be balls that are connected to another atom, another ball through this sort of sticks, and these will be all vibrating, and each of these balls will have a charge and will have a radii, and it's kind of but all of them are going to be super flexible, and it's just a matter of fitting this kind of a Lego construction thing, flexible Lego, into the active site. For example, you might recognize here this Coulombic term from your physics textbook. So that's charge, so something that's negative from the molecule and positive from the receptor, from the protein, will be interacting and will be enhancing the interaction between them. And you want something that binds in here, that will be the view of the, again, a drug or molecule binding into this active site. You want something that binds in there and it stays there for a long time. All right, so it will do its work. If something goes there and gets washed out in minutes, then you need to take the medication all the time. So you want something that goes there with very few quantity of drug. The more, we know that everything at some concentration will become poison. So something that has a small concentration that will bind very efficiently and will make it work. So with these mathematical formulas, you can now interrogate. Make simulation like this ones. This is a very fancy simulation. This is one of these receptors and a candidate molecule that's outside, and how this molecule moves around stochastically, tries different things, and the active site is inside of the protein, and we are showing actually one molecule in the active site, although it's not there in the simulation, and we can see how our candidate drug, and here we have a close up, does indeed find the active site and can enter. So that's super fancy. And that tells you how powerful are these molecular modeling techniques. So we call them techniques or molecular modeling techniques. How powerful they are, and how well they can describe this interaction. And if you could do this for every single molecule and try all of them, we would get very, very good drugs. But running such a type of calculation is very expensive. It is something that takes hours. In this particular case, it took around 24 hours, and it took around 200 computing cores to get that. So we can do it, but it's very expensive. We have much cheaper alternatives. So for example, this again is a protein. This is the active site, and we can make a grid of the active site. And in this grid, we assign each point of this grid sort of a charge, sort of we have also the three-dimensional shape of the active site with its concavity, convexity, with its corners, etc. And now we can try to fit all the molecules one by one in a much faster way, where the receptor doesn't move. The receptor is kind of rigid. The molecule might have a little bit of flexibility, and applying a reduced scoring function. So applying a much faster energy term, not the one that you were saying with all these energy terms. I think it's something simpler. And when we do that, it takes about 1 to 5 seconds per ligand. So we went from 48 hours or 24 hours, hundreds of computing cores, to a few seconds, one computing core. And well, that's great, but still it takes about 10 to 50 days if you use one computing core, or CPU here maybe it should be computing core, for 1 million ligands. And I'm telling you, you need many million ligands to find good molecules, because it's very difficult to find such a molecule. So it's still very complex, it's still very demanding, and that's why it take traditionally so long to take the thing. And you can use something in between, a little bit of just looking in the active site, a little bit of flexibility, few minutes. So there is a gradient of molecular modeling techniques with more or less computational demands that will do the job for you. So typically we apply hierarchical. So you apply something that's really cheap at the top level, and then you increase this complexity level, and at the end you for very few molecules. So the first one you start millions, then you reduce to hundreds of thousands, and tens of thousands, and few hundreds. And every time that you do that, you get you do a little bit better technology, a little bit better modeling technology. And so typically before AI, before machine learning, before another ingredients I'm going to be telling you soon, we could do around 1 to 5 million screening compounds on average. Of course, some people did 20 millions one day, it depends. And we got hit rates of 2 to 5%. So if we tested 100 molecules experimentally, we could get maybe 5 of them that were good or partly good. They were not excellent, and that wasn't a good day, right? So that was not the norm. So That's why, again, it took 2 to 5 years, very long time to find a good molecule. Then we have different techniques, which are kind of the precursor in that discovery of current machine learning techniques, which are the QSAR techniques, quantitative structure activity relationship. So here you don't look at the 3D receptor, you only look at a collection of molecules. So data, you start seeing this concept of data. You look at a collection of molecules that are known to be inhibitors for this molecule, for this target, or for similar targets, but you want something new because none of them is that great, or because you are a new company and you want to patent your own and bypass the others. So you can break down these molecules to smaller components, and these molecules will have a measure activity. And now you can make a mathematical model based on those components of each of these molecules and try to predict any of the molecule if it will be good or bad, just based on that kind of two-dimensional analysis, or we call it 2D, but yeah, it's basically a 2D analysis of the data space, of the previously existing data space of interacting molecules. That's faster, that goes significantly under a second, so you can do millions, much more millions than before, but typically the accuracy is much lower than the force field, these molecular modeling techniques that I showed you before. So, and now we go into what has changed the game. So what has been the revolution? The revolution here has been two main ingredients. One, the creation, companies like Enamine, which is actually in Ukraine. It's a company that's still working under the war and performing an excellent service, providing lots of molecules to many of our clients. So Enamine has been created Enamine and other companies have been created what it's called, it's known on-demand molecular libraries. So these are virtual libraries that they do not exist. So they don't have a molecule yet for each of these, they have not synthesized a molecule for each of these molecules in the libraries, for just entry in this library, but they know they can do it because these libraries were created based on chemical reactions. So now, currently, the real Enamine library has around 6 billion molecules. So imagine we said we will screen typically 5 to 10, 20 millions on a good day, and now these molecules have 6 billion. So thousands of times more, around 1,000 times more. And you know that the molecules that will be coming from here, you can have them in a couple or 3 weeks. You can buy them directly, but they will synthesize to you. They probably cannot synthesize 100% of them, but around 80% of them they will synthesize. And they are not expensive because these are they are based on building blocks, very easy to make molecules. And then, of course, what can we do? I mean, how can we search? We say we can do 5 million on a good day. So how can we search 6 billion? So, of course, the answer is machine learning. That's the only fast thing, not only machine learning plus data science, good structuring of these libraries, good handling of these libraries. But machine learning alone with this size of 6 billion, it handles pretty well. They are announcing, by the way, a 60 billion library for coming soon, and they are talking about a trillion, so 1,000 billions, a trillion library of pretty much all the feasible chemical space of molecules in the near future. So now we need searching technique. So you can say, okay, well, we can still make these models, and then we can search with much faster algorithms, algorithms that can break down these molecules in a non-linear way, in a much deeply analysis, much thorough analysis, and we can search those. We can now put these models of machine learning to search these libraries pretty quick. Then you can do with the current algorithms using 3-4 GPUs, you can do the 6 billion in 3 hours. So that's amazing, and that's how a lot of companies in the last 3-4 years got tons of investments, got hundreds of millions of investment to develop these things. However, machine learning standalone techniques have not provided an answer yet, have not really provided what they were being asked for. And actually, we have some of these companies that are using the last few hundreds of millions they have left to to buy molecules from some other way. So anyway, that's another story. But in many conferences in the last years, they have raised the concern of that these standalone machine learning techniques are not working. So machine learning alone cannot really provide an answer, cannot really find new molecules that are going to be good binders. And my 5-cent answer is that machine learning is good, it's very good, but good enough is not enough for this field. In this field, it has to be perfect because it's an extremely complex problem, and good enough is not enough. And there is the applicability domain problem. Maybe we can go backwards one more. So in machine learning, you will provide something new that's going to be similar to your training data. The applicability domain is critical in this field because you want things that are probably, if it were so good enough, you probably wouldn't have to find a new molecule, or if it's so close enough, it's not good if you're trying to make a new pattern. So it has to be something new, something quite different, something that will come out of the training data quite a lot. And of course, we know that AI techniques suffer from this applicability domain. So how can we break this applicability domain? And this is something we started working a year ago, and it works amazingly beautiful. And we are not the only ones. Many people are doing similar things. And every time I meet with some of these people, it's the same answer, same message from all of them. So you can use several AI techniques like screening, just screening using your machine learning model, or you can use generative molecules, generative methods like GANs or variational autoencoders to generate new molecules. Now, these new molecules, we know they suffer from applicability domain, but you can select a subset of these new generated molecules, and now you can use molecular modeling, the traditional molecular modeling, to interrogate, are these molecules as good as my machine learning model is predicting? And I can tell you they are not. So now this information from the molecular modeling, it's being used for active learning. All right, you pass the machine learning model. Okay, you said this is good, this is not good. Come on, learn from it. And that thing actually you predicted to be pretty bad, it turns to be quite good. So we interrogate both things: the things that they say to be very bad, and the things that it says to be really good. And we pass this information into the machine learning model. I can tell you that when you do that, you get hit rates. So out of 100 molecules that you try, you get around 50 to 70, up to 70% of true hits, which is a true revolution in drug design. How do we in practice do that? So for example, when we do screen the real enamine library, 6 billion, we basically take a clusterization of the library, and we take a subset. So each cluster, we take 1 or 2 molecules, and so that's a small space, 50,000 to 100,000 molecules. You can see a big reduction. And now we do molecular modeling, virtual screening. So we rank all these molecules, we select a subset of those, and with this subset, we create what we call a deep surrogate model, a machine learning model that will try to reproduce the scores, the energy functions that are coming from this formula I showed you before, for example. And then with this very fast and quick function, now you explore the entire library. You explore the entire library, that model will rank the molecules of the library. Those ranked molecules, you select again the top 50,000, you do again the virtual screening, and you tell him, no, you did wrong again here. So please learn from this, learn from that. And you do that in 3, 4, 5 iterations until you see that more or less you are converging. That's kind of the results that you get. What we see here in the x-axis is that energy function, that scoring. And when it's very negative, it's a very good thing. So what we want is a lot of molecules in very negatives, in this kind of red box here. And what we see in different colors is each iteration of this procedure that I show you. The first one is just that little guy over there with very, very few molecules with good score, which is what you would expect in a screening effort of 5 million, 10 million, which is what brings you to 3 to 5 years for finding a good molecule, etc. But now when you apply this hierarchical technique, you get, as you can see, the second one gets better, third one gets much better, fourth, fifth one, and you are increasing a lot the number of good molecules. So we could provide, for example, to our client, 86 molecules better than -11, which basically we didn't have any in the first iteration. And out of 86 molecules that had very good predicted virtual scores, 64 of them were hits. So, I mean, unprecedented. This is the best thing we have produced in our company or in our lab ever, and it was produced in the last 6 months due to this thing. And consistently in every project we apply this methodology, we get very similar enrichment factors. Here another example, and you can see here actually how you shift in each iteration. It's even more drastic how you shift for another system into much better scores. And here is a 2D representation of the chemical space that you keep exploring. So that's another great thing. Not only you get a lot of hits, but those are very different because you can enforce in your search to not retrieve similar molecules. So you tell the machine learning model, do not please give me always the same molecule. Give me different things because I want 20 different molecules to go into my lead optimization, to go into my final preclinical studies, animal studies, etc. So you can see how you cover a lot of the chemical space, much more than you would do in traditional virtual screening. So again, fascinating. And what is even more fascinating is the time. It takes about 2 to 3 hours for the screening. Of course, this is highly dependent on how much computational power you have. Here we use around few GPUs, not too many, 2 to 4 GPUs, but if you use more GPUs, you will reduce that. It takes about 6 to 12 hours for the molecular modeling, just simple, the one that took about 1 to 5 seconds, just simple virtual screening. And it takes about less than an hour for the machine learning reinforcement, etc. Altogether, one cycle in 24 hours. A little bit of human inspection, a little bit of cleanup. In 1 to 2 weeks, we do 4 or 5 cycles. In a couple of weeks, we deliver to our clients molecules that it took 6 months beforehand. Not one, we deliver a lot of molecules, a lot of quality molecules, something that took about 6 months before. So that's what I'm saying. It's a revolution. And as far as you have a target, as far as you have a structure, in principle, you can do it for every single target. Another idea that we play around is with generative models. So using, I mean, all of you probably are aware of them. So not searching in a library of molecules, but creating your own molecules, using generative models to create your own molecules. So the sky is the limit here. You know? We have an infinite molecular space that we can interrogate. A little bit of how we do this, so let's probably browse quickly about that, but basically you have a general set of molecules, data set like Campbell that has thousands and hundreds of thousands of molecules. Then you have your specific set, molecules that are known to be good for your target. Maybe you have some, maybe you have less, depends. If not, you will enrich that. And then you go into the machine learning, decoder, encoder, decoder to generate new molecules. And now these new molecules will go into an inner loop cycle where we will be interrogating for QED, which is the quality of the molecule as a potential drug. It tells you this molecule can be a good drug. So there are some indicators whether a molecule can be a good drug or not. SA, which means synthesis accessibility. Can we make this molecule? We are making new molecules. Can we pass it to our organic chemistry and he can synthesize it? No, that's a huge problem in this kind. So we do this kind of inner loop just to make sure you're making better and better and better molecules. We do this inner loop typically around 10 times. And we can also enforce similarity. So molecules be different from everything else that our competitors have been developing. Once we are happy with the quality of molecules we produce, we go into the molecular modeling. Again, we do this virtual, this docking, these things that took 1 to 5 seconds to get an estimate of the interaction between the ligand and the receptor. And then we tell the machine learning, these molecules are good, these molecules are bad. Start again. So again, it's this iterative process. And then at the end, the few hundreds of molecules that we are generating, we can go into some more strict filters, some more sophisticated molecular modeling, like the ones that you saw where moving everything around, they having this dynamical component, and then we can find our candidate inhibitors. So here, for example, this first picture on the top left corner shows the evolution of how much in these inner loops the model is learning how to make molecules. So it's learning quite well how to make molecules. that, of course, for example, you see a drop, meaning that they are not synthesis accessible or they are not good drug lines by the filters we had. Here you see, for example, the outcome of the molecules at the last iteration have even better synthesis accessibility than the initial training set, which is in darker. So we are developing molecules that we believe can really be synthesized. And here we have the scoring, the same thing we saw before, how good these molecules will be for our target. First round, second round, third, fourth, fifth round. You are starting converging at some point, but you are still increasing. So you are producing a lot of molecules that are good scoring, a good match. Again, a lot of diversity. In orange, you see the crystallized molecules space. All the crystals for this particular target that we did can be resumed in this 2D plot in that little space. Now, in each iteration, we are increasing significantly like that. So we can really enforce molecules that will be, for example, patentable. So will come out of anything else that has been produced before, but will be as good as what has been produced before, or even better. And just some more plots, for example, to say that we were capable of producing around 300 molecules, new molecules, with a Tanimoto index below 0.3. Tanimoto index shows you how similar are these molecules. So this Tanimoto was compared to the other 1,000 molecules that were described for this particular target, and we saw, you know, by going below 0.3, we are enforcing that they are very different. So 300 of these molecules. Then we went through a little bit more stringent molecular modeling techniques, and basically we got estimates of these molecules being nanomolar, which means basically they are very, very good binders. All right. So as you can see, here is maybe a good time to have a little bit of a break and to recap. So we are talking about a very complex problem that entitles huge amounts of money and huge delay in times, where good enough is not enough, where we need exceptional tools, where we need perfection, because otherwise people die, because otherwise you know, it's a disaster. And two things: the availability of big data and availability or teaching AI, machine learning, how to do a good job is critical. And with those things, we get unprecedented hit rates, something that has never been seen before. And we can do that in around 2 to 3 weeks. So it's extremely fast. So we can do several of these runs where we have a run of in silico, 2 to 3 weeks, a round of experimental validation, a round of refinement, etc., etc. So once we find a good molecule, then we are trying to optimize it. So now you find a molecule that could be a good guy, and now you try to make it even more perfect. And to do that, of course, we are also using a lot of molecular machine learning techniques. We are calling it a fragment artificial intelligence approach. So now you can imagine this is a representation of the molecule, this green molecule in here, inside the cavity. And for each of these cavity, we have different fragments. So now we are going to be telling AI, all these fragments are good to go in this particular cavity. Now generate new fragments that can even go better. So how we do that? Again, by virtual data enrichment. So we are going to be generating virtual data. We are going to be doing a screening, virtual docking of a lot of compounds. Now we are going to be fragmenting these compounds into all these subcavities that you saw before. So each cavity will have different set of fragments. And now we are going to be passing all these fragments to the machine learning. We're going to be creating a generative modeling with a general fragment list and a specific fragment list for our particular target. Generative model, again, a cheap level of molecular modeling reinforcement to pass the machine learning an active learning procedure. It's going to be making better and better fragments. And after that, we can build these fragments back into our molecule, and we can make fancy things like molecular modeling, fancy things like growing these fragments using this kind of expensive simulation force fields that might take half an hour to 1 hour. That you're going to be doing for a subset of fragments that have been highly optimized from machine learning, from artificial intelligence. And of course, we can use standalone AI techniques that will be using a lot of data to predict toxicity, solubility, administration properties of drugs. So of course, for that, machine learning is really good, and it can help you a lot. So that's also highly influencing and helping in bringing these molecules into clinical trials as much safe and as much performing well as possible. So we go from this 90% of failure in clinical trials to 17% of failure, to 60% of failure, to 50% of failure, and we will reach to this number soon enough, I believe. So talking about clinical phases, I mean, I'm not going to be talking too much about it, but AI will make a huge impact in the near future as well, and that's going to be coming from the digital twin. So we probably won't see this, but unless you are very young, but people will have a digital twin that will have a lot of your genomic information, a lot of your particular pathways that happen in your cells, and how the different drugs might affect you. And that will be not just for you to provide you better medication, to provide you personalized medicine, but we will have enough representative set of those that we could do the trials of new molecules first on these digital persons. And then we probably will be able to we are going to go for quite a while into real clinical phases, obviously, but we will discard a lot of drugs probably coming off these digital twins. Companies are not going to spend hundreds of millions if the digital twin tell them that most likely this will fail. So I'm going to just talk the last 10 minutes about other applications of this kind of active learning techniques in biotechnology. So other things that we are doing in the lab that we doing at BSc, we've seen this problem that we have a receptor, a target, which is a protein or enzyme, and how we are looking for a ligand, a drug that's basically a molecule that will interact with this protein. And that's very important for drug discovery. But the same mechanism of knowing this interaction can be important for altering the protein. So we can modify the protein, we can engineer this protein, and that can be good, for example, for enzyme engineering, to make enzymes that will work better. For example, now there are enzymes that work in industrial processes, and that using enzymes allows for a lot of chemical polluting processes to be changed into biotechnological, into green applications, where now we are not being polluting. We are developing metals. We can work at room temperature, not heating up, a lot of energy savings, etc. Or, for example, in biomedicine, for engineering proteins that can be work in the immunologic system as antibodies, etc. So molecular modeling certainly has made a big impact in the last few years. And we can, for example, interrogate how a substrate, in this movie we can see how a substrate binds to this enzyme that does a particular interaction, for example, breaking this molecule into two, and how we can enhance this enzyme, how can we engineer this enzyme, for example, to have a new catalytic trial, a new catalytic active site that will do more chemistry, that will do more performance. So this is the things that we do with these molecular modeling techniques, very similar to what you saw of that ligand diffusing and entering into the nuclear hormone receptor, that fancy simulation. But here now, what we are going to be doing is we're going to be changing the amino acids. We're going to be doing mutations. We're going to be changing these things to insert better performance or to insert additional catalytic triads, additional active sites that will be doing the chemistry. So now, once we have engineered the enzyme, we can check that the enzyme still works, and we can, in this case, produce basically optimal distances, optimal rearrangement of the active site to produce chemistry. So this, for example, has allowed us in the last year to engineer a nanopore, a protein that in the membrane of the cells that has a huge tunnel that can circulate large particles through it. We have engineered that to chew, to really eat nanoparticles of plastic. So this guy degrades plastic like no one has seen before. And actually, this is hopefully will be appearing in Nature Catalysis in the next month. We are right now at the third round of revision. So basically, we do that by inserting these catalytic triads in this pore, and being, because this spore is an octahedral, so we have 8 catalytic triads. We have like 8 scissors that are cutting plastic in any direction that the nanoparticle wants to adopt. And here you see the experimental. On the left, you see different nanoparticles, and actually the ones on the bottom are coming from a Granini plastic bottle from the lab, from downstairs the lab. So very custom plastic nanoparticles that can be in the environment, and after treatment with this protein, basically all the nanoparticles are gone. So, you know, enzyme engineering, I could say, will be critical for us as a species to be living in the next years. Not only, of course, for cleaning up the pollution, but for absorbing CO2, for reverting global warming, to doing all these things. So we need to do 2 aspects. We need to find enzymes that will work better, and we need to engineer those enzymes to do it. And of course, AI will play a tremendous role in here, and in our hands, that will be translated again into active learning. AI alone, and here we have even less data that we have in pharmacology for drug design. So we have even less data for this kind of applications. So active learning, using virtual data to teach machine learning, to teach AI how to do the job is very important. So how are we doing these things? For example, here we have one system where we had a lot of data. We had a lot of data. Of course, the way we are doing it is by using molecular modeling again, by interrogating whether the predictions are good enough, by telling the machine learning your prediction is good or bad. So for example, here we have one example where we wanted to find enzymes that were promiscuous. Promiscuous meaning that they could do the job for multiple substrates. These are esterases. They basically break down esters, and some enzymes, like the ones that you see in this part here, can break down up to 70 esters. So they are very promiscuous, this top part in here. And some other enzymes can only hydrolyze few tens or even less than 10 esters, and those are specific enzymes. They act on specific substrates and they are not promiscuous. But that was a very comprehensive set of data. Around 150 enzymes, cross-assayed with 96 different substrates, and you know, a lot of comprehensive data in here. So we built a machine learning model. Can machine learning, when we have all these data, produce a good model that we can find a new enzyme that will be promiscuous? And the answer is yes. So how did we do? We do first, as you do in any machine learning project, you look at your data and you do feature extraction. So we extract features from our data, and one of the main features that was important was the residue conservation. So all the enzymes have basically this amino acid sequence, and you can map how much an amino acid is being conserved. For example, in this position here, this R with an arginine, all the proteins have this R, so it's a highly conserved amino acid. So this conservation maps was very important. It was a feature that we extracted from all this list of experimentally assayed enzymes. Then we built a model. Still, while for enzyme engineering this is a lot, this is one of the huge data sets that we have, still this is nothing for machine learning. It's nothing for AI. So basically we use 3 different techniques that are capable of handling very sparse, very few data. Nearest neighbor, support by machine, risk classifiers, and we actually did a consensus ensemble of these guys, and we got actually very good MCC, Matthew correlation, that can separate promiscuous from non-promiscuous, 0.8. We were pretty happy. Then we put this thing into work. We went into a huge database of enzymes, 280,000 enzymes. We run our model. We selected now things, and then we run them with our molecular modeling technique to see which one we could set. And actually, we saw that there were some of them that were predicted by our molecular modeling techniques as highly promiscuous. So we went ahead and we told experimental people to try these 10 sequences, and indeed those 10 sequences came out to be very promiscuous. So if you have all these data, you can do a lot of machine learning, and you can do, you can buy a prospect. You can go now into metagenome data sets, and you can find enzymes that will be doing good work for you. And what about if we do not have the data? So that's That's what we are working now. If you do not have the data, you have a sequence of space of enzymes, but you don't know whether those enzymes are good or bad. Now you make network analysis. So you put the enzymes into groups. So which one is good, which one is bad. You create the structures, you do molecular modeling. Now you test it and you tell the machine learning what you expect from molecular modeling to be good or bad. So use virtual training to tell the machine learning and now create models to the bioprospecting. And now we are doing that in several projects. We are using around 1,000 to 2,000 molecular modeling simulation. Each molecular modeling simulation turns into all these kind of maps of protein substrate interaction. We can predict which enzymes are going to be good, which enzymes are going to be bad. And now we're working to translating that into machine learning models and to tell the machine learning model to bioprospect large genome or metagenome sequences libraries. And the last thing I want to tell you is similar things you can do now with generative modeling. You can use generative modeling to create new enzymes, and that's really hot. It's being on the news, and it seems to work quite well. So you can start with some sequences. Now you can make your variation to coder or your GANs to generate new sequences. Now, because AlphaFold, you can get the structure of the sequences. Now you can test molecular modeling to see whether those sequences, enzymes, are doing what you were expecting or not. You can pass this information, and again, you can iterate and make it better and better and better. And for example, we did that for one client, a little bit more complex procedure, but at the bottom line, experimentally, it takes about 6 months for them. They say 100,000 sequences, very expensive. They didn't tell us, but I can tell you it's super expensive. And they resolved with 12 sequences with good binding affinity. It took about a couple of months for us. Now we can do it probably in a few weeks. We assay virtually 135 virtual proteins. 12 of them were tested experimentally, and 7 of them were well expressed, and 2 of them were having very good affinities, were resulting. So, as you can say, huge advantage in respect to experimental techniques. So let's finish here. And
okay,
here is the conclusion. Yeah.
I don't know if you can. I know that you are finishing your speech.
That's the last slide. Yeah.
Is the last one? Okay, you can finish, and after that we can start with questions. Thank you.
Yeah, it's just the last one. It's just basically summarizing. You know, HPC and artificial intelligence is changing. It's changing the way we look at things. Is opening so many doors. Still, standalone techniques, standalone machine learning techniques are not good enough, and maybe are good enough, but they are not perfect as we need to be. I have no doubt they will be in 10 years when more data is available, but today we have to produce active learning of those. We need to reinforce them, and we are speeding up so much these things that we can envision in 3 years having this virtual platform where the scientists will interact with these modeling techniques in real time, and that will boost the mind of the scientists. The mind of the scientists will fit directly the ChatGPT-10 model that we'll be using at that time, and everybody will be making it stronger and much faster for solving very difficult problems. So I'll stop it here, and I'll thank you for your attention. I'll thank people at BSc and at also by Discovery, which are the real ones who are conducting all these studies.
Okay, so thank you, Victor, and thanks for your interesting speech, and congrats for your hard work on molecular modeling research line. As said, one of the attendees has a question for you. This question is about if you could explain, in case it's possible, not confidential, the architecture of cloud and supercomputing used in all of these artificial intelligence process. As you said, faster. If I don't know if it's more cloud investment or more supercomputer investment that you are needing for your research.
We use Amazon Cloud. We use sometimes we have some projects in collaboration with BSc, so we use as well Mare Nostrum sometimes if we have a project that we are awarded, and so we use what everybody else is using. We use even our cluster. We have an internal cluster, so you don't have to have the Google supercomputer. You can do it with small resources. For example, a full 2 or 3 week study on these things will probably cost you in cloud computing 3,000 to 5,000, no more than that. And you know, it will provide you this amazing hit rates in a fast manner. So it's certainly not expensive. And if you are willing to spend a little bit more weeks, you can make it a little bit slower. You can reduce the cost significantly. So it's HPC, so you cannot do it in a laptop. But we are not talking about requiring the OpenAI resources. No.
Mm-hmm. Okay, thank you, Victor. I have another question. From the beginning of your presentation, you told us that to find a molecule in a classical way, okay, takes no more than 3 or 5 years to achieve this molecular as a needed for a drug or something. And finding a molecule using this modeling and machine learning tools developed by your group and in this new modeling research lines, it's about 24 hours more or less. So my question is, how about the feedback coming from pharma sector? Is pharma sector ready to start working with these new tools, or how about it?
So let me be more precise. One cycle takes around 24 hours. From starting your project, when you have your target validated, to get some molecules that can really go to start some serious animal models or clinicals, it will take 2 to 3 years, right? Traditionally wise, somehow. We can now reduce it to 4 or 5 months. That's the bottom line, because it will take us 2 or 3 weeks for the first set of molecules. Let's say 1 month for experimental validation, or 2 or 3 weeks more, then another 2 to 3 weeks for optimization. So now it takes us around 4 to 5 months. So we have reduced it from 2 to 3 years to 4 to 5 months, not to 24 hours. And the industry is excited about it. I mean, we are applying it into, you know, with many pharmaceutical companies, and we are not the only ones. I mean, I want to leave this clear. I was just talking to a colleague of mine that has another company in Barcelona, Xavi Barril, and he was showing us his results last week. They are exceptional as well. And he was saying, I mean, this is a revolution. This is a completely game changer. It's unbelievable. So it's not that we have the truth and the others don't. It's this kind of techniques, when they are properly used, they can produce these successful results.
Okay, Victor. So let me ask you another question, more or less based on that. If you are speeding this procedure of finding a new molecule, let's say for being using this drug discovery procedure, how about the certification procedure? I don't know the technical word, you know, this, so to be, if this molecule is being used in a new drug, let's say, you are cutting or you are speeding the molecular procedure so significantly, but I guess that the certification procedure takes a lot of time as well. So how to balance it? It
is. So we are speeding up those 3 years to 6 months, let's say, to put some number, or those 2 years to 6 months. So we are making it like 4 times faster, at least. But the nice part of it is that the molecules that we are producing are much, much better. And we are adding a lot of diversity, a lot of control at these early phases that you could not do before, because you were lucky enough before to get a good molecule. So, you know, then you had to do a lot of optimization. Now you get so many good molecules that a good medicinal chemist will get his work, you know, highly advanced already. So you still have to do all the procedural certification, certainly all the clinical phases, etc. But those are going to be much faster. And those are going to be, we would expect a much better results. So they're going to be, of course, they will have a limit constraint, but the results coming of them will be much better. You will get a lot more success rate in the molecules that are coming. Of course, we just developed this for the last year in general, our companies. So we are going to see in the next clinical trials in the next 3, 4, 5 years how these new molecules being of superior quality have really improved the success rate. This is coming in the next years.
Okay, thank you, Victor. I guess that we have another question, so I'm not sure to. One moment. I'm not sure if I can see the whole question, but it's about this generative artificial intelligence give information only on third or fourth hinge structure, or can also learn on toxicity and other main characteristic to obtain human tolerable molecules. Yeah, that's a nice. I really like this generative modeling approach. I think I like both, and I think what we are seeing is that for many of the systems, with 6 billion, you get so many hits. You can screen 6 billion molecules, and there are good molecules. There's a lot of them. But in some very difficult cases, and as the attendee says, where you want to bring a lot of limitation factors, generative models allow you to do that. If you have some filtering functions, if you have something that can more or less correlate what you are looking for, and in that case, QED filters, synthesis accessibility filters, but you could bring CNS filters. Well, you can bring a lot of different filters, ADMET filters that will drive the generative model towards your goal. And the good thing is that you just keep running it. It's fast enough. You keep running it until something good appears. So it can access the entire space of molecules. So it's perfect. So yeah, the only bottle problem there is that then you will need to have some synthesis. So far, we are seeing that the molecules coming out of them have good synthetic routes, but yeah, that's the only problem, that it might take you maybe 6 months to really make the synthesis of that molecule. So therefore, it's a trade-off of what you're searching. And then, for example, another thing we are implementing is that we are using the generative models. We are making lots of good molecules with generative models, but then we are looking those molecules to search in the large space of the real enamine for something that already exists, or something that's very easy to make, that will be looking to those molecules. So just make your wish list in generative modeling, and now use your wish list to go into one of these huge on-demand libraries to search for something that will be looking like, and that can do still the same work. So that's a hybrid method that we are also implementing. Okay, thank you, Victor. Another question is about, you are saying that you are needing a huge database for this machine learning procedure, okay? The question is about where this data is coming from, who are the owners of this data, on how to proceed with this.
So that's one of the beauties. That's one of the beauties of what I'm trying to show today here. We might not even need data anymore. We can produce virtual data that's good enough to. We can start the simulation now with 5 binders, or even without any binder. We just do virtual screening. We select the best predicted guys of virtual screening as the good guys. We assume that that's my initial data. And now I start iterating. And what I'm going to be doing is better and better and better data all the time. And I know that when I have a lot of good results, a lot of good guys predicted to be good, the success rate, the success probability there is huge. So now we advocate that you don't even need real data anymore. And I think machine learning in the next years will not need any real data anymore. It will work only on own generated data.
And how about this virtual data that you are generating? It's
you can generate it on the flight. You can. Yeah, the good thing is that you can generate it on the flight. It's pretty fast. So you can, of course, you can store it, and you can build your own database that belongs only to you. Of course, here for the problems I've been telling you. Now, if you go to target validation, to other problems, or digital twins, or then there is a huge problem, which is, patients' records, public medicine data. That's a different story, and that's a real problem. And anonymization might be a solution, but here is a lot of sensitivities, and I'm talking more about the virtual screening. That my idea is that we won't need any real data soon.
You are producing your own data.
Exactly.
Based on
that data is bad at the beginning, but it gets, by this iterative process, it gets improving, and at the end, it's much. You turn this bad data into excellent data.
Okay, and if you are able to create this virtual data in order to, I don't know, it is an open question, in order to speed up the certification procedure, I don't know if you can consider as well to generate in some way this virtual patient in order to.
Well, those are the digital twins, yeah. That will take some time, because understanding well the metabolic pathways in a cell is very complex. It will happen, I have no doubt, but not in the next 10 years. It will happen a little bit later on. And at the beginning, so of course, no one will believe them, or very few people will believe them. They will be good for classification. So don't try this drug, try these others. And later on, maybe they substitute real clinical assays. Probably that's going to happen.
Okay, so based on the application, I guess that we don't have any more questions there. So I don't know if the attendees wants to make, so they have time to make other questions. I don't know.
Anna, sorry, there are 3 more questions, but I don't see if you can scroll, because they are like going down one to the other.
Well,
there are
you,
5. Yeah, thank you. The third one would be the next one.
I cannot see them. That's I don't know.
Okay, you can. I think you can enlarge the window, but if not, if you want, I can ask the question to Victor if you can find
because, it. Yeah, I have this. Okay. I have done this, but not able to manage the. I only can see one question.
Okay, I'm sorry to hear that. Okay, so Victor, the third question would be about the type of analysis that you explain in your presentation. There is one attendant who is asking if it's very expensive, this type of analysis.
It depends. It depends what you want. So, for example, I can tell you from our company, I cannot talk about the others, but our company, a full virtual screening problem, like finding molecules for a target. Again, as I told you, if I take 2 to 3 months, that would be on the order of 60 to 70,000 euros, something like that, overall, which is a fraction of what it will cost experimentally, which we'll be talking about several millions of euros. And while 3 years ago, that basically was the same price, but we get 2 or 5% hits, which still were good. But now we get these 50% hits, so now we can almost guarantee good hits. So that's in that sense, it's very attractive for the industry.
Okay, thank you. Another question would be, what do you think are the main challenges of this technology in the next few years and entry barriers that you are facing?
The next challenge, I couldn't hear that well.
Yeah, sorry. What do you think are the main challenges of this technology in the next years and entry barriers that you are facing?
So the title is not a joke. I think finding a molecule for a target is a solved problem. I believe it. I mean, I have plenty of confidence in that. So now, to me, the problem is okay. In one side, in the drug discovery is the target validation. So finding good targets and having targets different enough from the others that can bring you this competitive advantage. If we are talking about the technology we have been showing for virtual screening, what are we working on is to make it faster, to make it more robust, to make it cheaper. We have, I do at least personally have a big compromise with environmental issues. As you see, we are trying to really come out with proteins to degrade plastic, but global warming for me is a big issue. So I want to use less computational power. I want to burn less electricity to do these things if possible. So at least for me in particular, that's one of my targets. But make it more efficient, and what I show you at the end, that I want to make it fast, so fast that I can give people a virtual tool that they can really do their own designs based on their experience. Maybe they can hire this guy in Japan who's an expert of this particular enzyme and have these virtual sessions with them and create new molecules on the flight. That's what I would like to work with as well.
There is another question, the last one, if there is anybody who is asking for another one. And the last one is thank you for your presentation. And the question is having a little introduction. He says, during your presentation and answers, you strongly rely on scoring functions that were developed many years ago to train the neural networks and reinforcement learning. And the question is, isn't there a need for improvement in those scoring functions? Are they still valid for drug design or enzyme design?
I think actually this will give them a second birth. In my hands, they are very valid because I'm trying it and I'm seeing that they predict on it. So what was the problem of the scoring function? The scoring functions, so it's a combination. Scoring functions, not being able to interrogate enough molecules, and being very poor at predicting medium range molecules. So if you have all your predicted molecules that are if you can imagine that for your molecule the best score will be -11, -12, in your traditional docking you will get maybe 1 or 2 of those, and then you will get hundreds of -9 and -8, which are just basically by default false positives. But the iterative procedure coupled to these massive libraries keep enriching the search, keep finding molecules of -11, -12, -11 that you could not find before. And with those molecules, the false positive rate is significantly lower. Certainly, if you can come out with the better the scoring function, the better you can predict how good it is by new scoring functions. Awesome, and we've been trying, for example, scoring functions coming from machine learning, and at least in our experience, I believe we try pretty much all of them. They are not good enough. The old traditional molecular modeling is better than those scoring functions, because here you're trying to interrogate molecules that are significantly different from what they've been trained with, in principle. So then they are not performing that well. We try Deep Dog, we try all these guys, and they're not yet there. And I have no doubt that they might be there at some point. Don't get me wrong, and that's why I say my conclusion: standalone AI techniques might be good enough, but now the combination of these old traditional scoring functions, where again I insist, you do this iterative procedure, so you keep enriching them, so you get the best of them. That's truly performing in my hands, in our hands.
Perfect. He says thank you for the answer. So that's
to attendees, please feel free to send me an email and contact me if you guys have questions. I will be super happy to answer any question or talk more, debate more about this field. So.
Okay, so I guess that we don't have any more questions, isn't it, Marta?
That is correct, Anna. This was the last question in the section.
Okay, so I guess that we can start finishing the masterclass. Victor, we are receiving a lot of thanks and wonderful masterclass, super interesting. So congrats for your speech, Victor, and your research line. And we want to remind you all the attendees that we are going to send contents and the link to the master's class that has been recorded. And the idea is to remind you that the next masterclass that CIDAI is going to organize is next month, the 16th of May. And the masterclass will be about generative artificial intelligence, and the masterclass will be led by NTT Data. So it's another interesting masterclass on artificial intelligence. We strongly recommend you to connect next month masterclass in order to have this interactive space in order to share these research results and innovative solutions in this artificial intelligence world, let's say. So thank you again to all of you for being here and to provide us these interesting results, this interesting feedback, and thanks again to Victor to providing us this fresh content about his research results. So I guess that we don't have any more questions waiting to be answered. So that's it. Thank you to all of you.
Thank you, everybody. Thanks, CIDAI.
Bye.